Good morning, friends. Today we will be talking about the modes of evolution. This lecture is about the first part of the mode of evolution. The next part would be covered in the second lecture. The mode of evolution is usually identified and studied by means of the fossils, and the geological time offers a very neat way, a very convenient way in which the patterns of the change and the speciation can be observed. And fossils provide a very unique opportunity to understand the change in morphologies, and the morphological change is also a kind of the change in the constraints in which the animal lives. When the constraints in which the animal, the physico-chemical constraints in which defines the habitat and ecology of that particular organism, when it changes, the morphological features also change. And such changes are like that it is very difficult to observe that in a very short span of time, because these changes are very long term. So, you need a very long geological time in which such changes are felt. The problem with the study of such transformations, the formation of different species, formation of different morphological features, formation of the big groups, and the big changes at the individual level, the species level, or even at the cellular level. These changes, they are subject matter of their recording into the stratigraphical record. That means, if they are preserved in the sediments, if the stratigraphical record affords to indicate them, then only such changes can be studied. Otherwise, it is very difficult to observe such changes, and it sometimes creates a lot of confusion as well. Confusion in the sense that any particular change is gradual. So, if I am getting a new form from one particular form, then there had been several intermediates also. So, if there can be two possibilities, one that were intermediates and then the final form was evolved from the initial form through a series of very gradual changes, and this is one category. In the other category, the new form evolves abruptly from the pre-existing form. These two categories have been very cordially named. The first category is called as phyletic gradualism, in which the changes are slow and gradual, and you obtain the final form from the initial through some very intermediate forms as well. But if you try to talk about the punctuated equilibrium, that is the second category, in which the new form is derived abruptly from the pre-existing form. Now, consider a case in which the intermediate forms are not preserved into the stratigraphical record. That means, if A evolves to B, B evolves to C, C evolves to D, and only if A and D is preserved, then one might think that this represents a punctuated equilibrium. This will be recorded as punctuated equilibrium into the stratigraphical record, but in reality it was phyletic gradualism. So, that is a kind of a function which involves many factors into the consideration. The two principal factors is that it should be happening into the individual level, and it should also get a chance to be recorded into the sediments. It should be preserved into the stratigraphical record. We will talk about two different modes in which the natural evolution is manifested. One is called as the microevolution, the other is macroevolution. Let us first talk about the microevolution. As far as the microevolution is concerned, microevolution is the evolution in which you produce the individual, that means it is at an individual level, the changes into an individual because of the ecological constraints is called as the microevolution. That means the force of such kind of the genetic change which is expressed as phenotype by different characters. So, such kind of the environmental driven changes reflected into the individual at the individual level is called as microevolution. So, this represents a small scale evolution, and the examples of microevolution is seen in the Cretaceous sea urchins like Micrastra is the best example of the microevolution, and in which we see morphological changes, but at the same time it has to be observed that this kind of the change has to happen over a wide geographical area, and if it is happening over wide geographical area, you will be having certain intermediate forms which attest the microevolutionary processes. But if these forms are not preserved, then it will be difficult to ascertain the microevolution which is understood in terms of the individual level changes. And fortunately, Micrastra is an example in which microevolutionary changes can be studied. But there have been many other microevolutions which happened on the earth, but did not get preserved into the stratigraphic record. So, that is a kind of a dilemma. And then sometimes the manner in which it occurs is allopatric speciation. What is allopatric speciation? Actually, allopatric speciation occurs when you have the geographical isolation. The daughter species, the offsprings, they become separated from the parents by some geographical constraint, and if because of the new environment they start to get new characters. However, they got split from the parent species because of the geographical isolation, but then they start getting new characters. This new characters leads to the reproductive isolation. That means, these two offsprings from the same parent species, they can no more interbreed. That means, they can be categorized into the different species. I mean to say that then the reproductive isolation makes the new species. The reason is the geographical isolation. This is particularly called as the allopatric speciation. When this reproductive isolation occurs without any geographical isolation, that is called as sympatric speciation. The speciation basically is of two different types. I hope this is something clear to your mind. Let us be confined to the allopatric speciation first. So, in the allopatric speciation, as I told you that it is because of the geographical isolation, geographical constraints. That means, the organism starts to adapt to a new setting, and when it adapts to the new setting, there may be changes in its size, there may be little changes in its phenotype based on the constraints which the environment imposes on its genotype. This also comes under the microevolution. Well, but we can say that all the individuals of this this entire offspring which got separated, this has been called as peripheral isolates by some people. So, among these peripheral isolates, you have the individuals that will survive the new constraint. You will have animals that will not survive this new constraint. There may be individuals which will adapt to the new environmental setting. There may be animals which cannot adapt. So, those who cannot adapt will become extinct, will be wiped away, and as that gets wiped away, you have some genetic information permanently lost. So, the new adaptation that will happen from the survivors which do not represent the exact genotype from which they were isolated. So, there is a chance of missing information, that means this manipulates the genotype, and this is something which is a constraint and cannot be avoided. So, this is what is called as the allopatric speciation and microevolution in terms of the allopatric speciation. Well, there can be a very subtle discussion which concludes this microevolutionary change, after which we will be discussing the macroevolution. The fact is that when the stratigraphical record is continuous, we find that there is no visible break into the sedimentary record. All the sediments were deposited, but the deposition of the sedimentation continuously is not a guarantee that it will preserve all the morphological features, all the events of change into the rock record. Why? The reason is that 1 millimeter layer in the sediment gets precipitated after 1000 years. It takes about minimum 1000 years for the general rate of precipitation to lay down a 1 millimeter thick layer, and 1000 years means almost 10 human generations. If one generation is considered to be worth 100 years, then 10 generations would constitute just 1 millimeter layer. That means, and after one layer, the second layer will be deposited immediately is not a guarantee. So, if there is a gap of 1000, 2000, 3000 years within from one layer to the other, that is something which go unnoticed into the sedimentary record, and that would be called as conformable and continuous. But on the biosphere, if you see that such a time gap, these small subtle time gaps which are principally called as diastems, they affect the changes occurring into the biosphere a lot, and these changes will not be recorded in these minor gaps. So, you have the sedimentary package consisting of the depositional gaps and depositions that interludes between that gap, and you cannot avoid the presence of minor gaps, which is diastems. So, the conclusion of this entire issue is that even if this stratigraphical record is continuous, it is not a guarantee that it will record every change that happens into the biosphere. With this, microevolution is over. Now, let us talk about the macroevolution. As far as the macroevolution is concerned, it is not essentially upscale version of the microevolution. Microevolution and macroevolution, they are two different categories, they are two different terms having different meanings. Macroevolution means the evolution that occurs at the species level. Microevolution refers to the changes that occurs at an individual level. So, this is one scale up, we have to admit that at this point. But if you try to see that the changes which is incorporated under the term macroevolution has the big scale changes. Like for example, earlier cells, they were without any nucleus. These cells were called as prokaryotes. Then you start to evolve from these cells that has a well defined nucleus. That means the change of one cell to the other, one without well defined nucleus, other with well defined nucleus, this is a big, I mean it is a small change occurs at a species level, but it can be transformed that entire eukaryotic evolution is based on this minor change. So, this will come under the macroevolution. After the Cambrian, you had the life exploded on earth. Many forms came with very complicated morphological features. You could see trilobites with well defined head, cephalon and shield with eyes in the Cambrian, and no feature in the Proterozoic or Precambrian. Suddenly they evolved, and this kind of a sudden appearance is quite anomalous, which is very popularly called as Cambrian evolution of life. The fact which I am trying to convey this to you is that such changes which have the broad scale effect are termed as macroevolutionary changes. The evolution of the monograptids from the diplograptids, that is something which comes under the term macroevolution, and most importantly the evolution of suture pattern into the cephalopods. And fortunately it was recorded and we could observe that. If you see the suture of a nautiloid, suture means in a chambered body, in a body in which the space is divided by septa into different chambers, the surface expression, the expression of that septa onto the external surface is called as suture. So, this suture basically is something which bears the hydrostatic pressure of the water column. If the cephalopod is shell is hanging in the water column, that means the hydrostatic pressure exerts some load onto the shell. So, the shell must be in a position to bear that load. So, force is something which cannot be reduced, force will be constant. So, if the organism changes the area, the organism can balance the pressure. If the area is increased, the pressure will be reduced. As you know that the pressure equals to force upon area. So, if you cannot change the force, by changing the area you can change the pressure. That means in spite of the fact that the force is considerably higher, the pressure becomes lower because the area increases. And the only way that these cephalopods have found out to increase the area is to complicate the suture. So, if you see the suture pattern into the nautiloids, it is very simple and slightly curved suture that you observe. But in ammonoids, the suture pattern complicates a lot. That means the curvature forms the kind of the lobes and saddles, and such lobe saddle type of the suture is preserved in a form which is called as ceratitis. See the suture of ceratitis, it is having the lobes and saddles, so little complicated suture. But then the complicacies they even modified further. Most of the Mesozoic cephalopod, especially ammonoids, if you see, they have a very complicated suture pattern. So, this suture pattern from the saddles and lobes, you see the saddles and lobes both become frilled, then this frill becomes, it forms the auxiliary lobes, sometimes it forms the adventitious lobes also. So, you have a huge complication of the suture pattern. The reason is to increase the surface area. Once they increase the surface area, the load is reduced. Why they wanted to reduce the load? Because they wanted to explore the other ecological niches, maybe for food or dominance. So, they explored themselves to the vast horizons because of this complicated suture pattern and the shell that became strong enough to hold that capacity to withstand the hydrostatic pressure. But an organism is having a limited range of pressure, temperature, the physico-chemical conditions in which I survive. If you suddenly reduce the temperature, I will die. If you suddenly increase the temperature, I will die. I have a very small range of temperature in which I find myself comfortable. But if I increase my ecological niche to which my body is not adapted to live, then ultimate fate is that it will become extinct, and yes, ammonoid became extinct. So, the food and dominance might have been the ecological constraint for this major macroevolutionary changes. Anyway, I have tried my best to explain you the mode of evolution in terms of macroevolution and microevolution, and some very excerpt examples from the paleontological record. I hope you might have understood it. We will be discussing the second part of the mode of evolution in the next lecture. Till then, thank you very much.